Right, so we are working on the citrate cycle. We're going to start with those enzymatic reactions that are going to drive the citrate cycle. So you have to remember that the citrate cycle is all about oxidation, right? We're going to oxidize our substrates and we're going to reduce our electron carriers. So really in the citrate cycle, we have 8 reactions, but we have 4 coupled redox reactions that are really going to transfer pairs of electrons from those organic compounds, those organic substrates, to those electron carriers of NAD plus and FAD. So you have to remember, right, we've got pyruvate. Pyruvate gets converted to acetyl CoA. So we've already produced 1 pair of electrons and sent them to NAD plus to create NADH. So if you're just talking about the citrate cycle, nothing else, if you look at all the reactions, what are we going to produce? We're going to produce 4 NADHs, right? We're going to produce 1 FADH2. We're going to produce 1 GTP, which basically is the same thing as ATP. So you can kind of think about it that way. But when we look at this cycle is an aerobic pathway, right? We need O2 in order to make it happen, because O2 is going to be our final electron carrier, and that's going to be really, really important to keep this cycle going. So this is a continuous cyclic cycle, right? It's the product of the reaction is going to become the first substrate, and we're going to keep going over and over again. So remember how I said there were 4 redox reactions. So you need to know which 4 of those reactions are redox reactions. So it is 3, 4, 6, and 8 that are our redox reactions. And if you look at this, how do you know that these are redox reactions? What are you producing? You're producing NADH, you're producing FADH2, and another NADH. So these are all reactions where you have redox occurring. So if you kind of look at the enzymes for these reactions, you can look at them and you can predict what kind of reaction it's going to be just from the name. And you can also kind of look at these and say, okay, which ones are going to be favorable and which ones are not. So if you look at our very first reaction with citrate synthase, this is going to be actually a condensation reaction. Aconitase, do you see how we go from citrate to isocitrate? You're just preparing the substrate to be ready for the next reactions. This one's not really, really that important. It's really just an isomerization reaction, right? Then the third one, when we go from isocitrate to alpha-ketoglutarate, and we produce that molecule of NADH, that's isocitrate dehydrogenase. And this is a really, really important one, because this is oxidative decarboxylation. So every time that you see a decarboxylation reaction, what you're going to be producing is also a molecule of CO2. So we have an oxidation reaction occurring, and we're also decarboxylating or removing a carbon. So those carbons come off as CO2, right? So look at the next reaction. We produce, again, NADH and CO2. So this is alpha-ketoglutarate dehydrogenase, because that's the substrate that we're converting to Succinyl Coa. So this is yet again another oxidative decarboxylation, right? Then in 5, right? So if you look, we're producing a GTP. So this is a phosphorylation, right? We're gonna take that inorganic phosphate and pop it onto GDP. Then 6 is succinate fumarate, and we make a molecule of FADH2. So you actually have an oxidation reduction reaction occurring there. Then fumarate to malate, all you're doing is really a hydration there. So you can see water coming in being important, so it's just a hydration. And then you go from malate to oxaloacetate, and that's our final reaction, because oxaloacetate, if you look at the top, is a substrate. So that's a redox reaction. So the one thing that's kind of important to be looking at here is which delta G values are really important, right? And we have some that are really, really negative, right, that are driving this reaction. And the rest are pretty much close to 0, except for one, this last one. This last one is pretty positive. But here's the deal with this one. Even though it's very positive, the substrates, the products here, the oxaloacetate and the NADH, these two are depleted so quickly that even though they have a positive delta G value, it still will drive the reaction forward. So it's because those two substrates are depleted really, really quickly, right? Okay, so remember that in this reaction, right, if O2 is limiting, right, if we don't have oxygen in order to be in a final electron carrier at the end of the electron transport chain, then NADH, FADH2 are going to accumulate. And then we're going to have some feedback inhibition, right? So just kind of keep regulation in mind as we work through these cycles. So if you kind of look, you can kind of simplify this reaction just by looking at exactly what's happening in the reaction. So if we look at pyruvate and we convert it to acetyl CoA using our pyruvate dehydrogenase complex, right? We start out with something that is basically a 2-carbon molecule. And we attach that 2-carbon molecule to a 4-carbon molecule. And in that very first reaction, all you're doing is you're tacking the 2-carbon molecule onto the 4-carbon molecule so that you end up with something that is a 6-carbon molecule. So you have one to erase, 1, 2, 3, 4, 5, 6, right? So we have 6 carbons there. And so if you look at that very first, reaction, you're just transferring that acetyl group to basically the oxaloacetate backbone. And when we go from reaction 1 to reaction 2, all we do is an isomerization. So you can look, you have 1, 2, 3, 4, 5, 6, right? We have 6 carbons inside of our chain. So we really haven't done too much. But now, when you go from isocitrate to alpha-ketoglutarate, this gets really important because what's going to happen here is if you count those carbons in this next one, we only have a 5-carbon chain, right? So we had one of those carbons that came off. So this is our oxidative decarboxylation. We're oxidating our substrate. We're reducing NADH, right? You can see that right here. You go from NAD plus to NADH, and we lost a carbon. So that's our oxidative decarboxylation. That's really, really important. Then we go from alpha-ketoglutarate to Succinyl Coa via alpha-ketoglutarate dehydrogenase. So you know this is going to be another oxidation reduction reaction. And so we take our NAD plus and we convert it to NADH. Now you go from Succinyl Coa. And this is oxidative decarboxylation again, right? Because you're losing CO2. So if you count your carbons, right, 1, 2, 3, 4, we've lost another carbon. Now, from here on out, once we make Succinyl Coa, we're not going to lose any more carbons, right? Because Succinyl Coa is a 4-carbon molecule. And we already talked about up here, oxaloacetate is a 4-carbon molecule. So you're not going to lose any more carbons after this point. So the next reaction, right, we're going to have our substrate level phosphorylation. You remember this from glycolysis, where we actually use the energy transfer to phosphorylate. In this case, it's not an ATP, it's a GTP that we're going to make. So we go from Succinyl Coa to succinate, and that process generates our GTP. Then we go from succinate to fumarate via succinate dehydrogenase. So you know what's happening here. We're taking our FAD and we are reducing it to FADH2, right? So our substrates get oxidized and our electron carriers get reduced. We make fumarate. Fumarase converts our fumarate to malate, and that's a condensation reaction. And then what we're going to do is we're going to convert our malate to our oxaloacetate via malate dehydrogenase. So we're going to make one more electron carrier molecule, right, that we're going to reduce. So you kind of just got to keep in mind what's happening in the citrate cycle. And the whole point of the citrate cycle is to generate these NADH and FADH2 molecules that are then going to go on to the electron transport chain. Yes, you're making one molecule of GTP, but that's not the really important part. The really important part is that we want to fuel the electron transport chain. Okay, so there's a really great animation that you should go and watch. You should absolutely go and watch it. All right, so let's go through these really step by step, right? So when we look at that very first reaction of citrate synthase, this reaction is irreversible. And how do we know it's irreversible? It's that large negative delta G value. So the purpose of this is really to commit our acetate unit of acetyl CoA to oxidative decarboxylation. So make sure you write that down. Purpose, we're really going to commit acetate unit to oxidative decarboxylation, right? So it's really a condensation reaction, and it's very highly favorable, right? We have a very large delta G value. But what we want to do is we really want to take those 2 carbon molecules that are in the acetyl CoA and commit them to this cycle. And so that's why we want this to be an irreversible reaction. We want this to go once we start, right? Okay. So the next one is, right, just simply an isomerization. And it's actually a 2-step mechanism. We write it in the cycle as only a 1-step, but it's actually a 2-step. And so you're going to convert citrate eventually to isocitrate. And so when you look at how this reaction works, really we have 4 iron sulfur clusters inside of our enzyme. And those iron sulfur clusters are there really to remove the hydroxyl. And so if we look at the conversion of citrate to cis aconitate, if we can look at what's actually happening, what we're trying to do here is we're trying to remove hydroxyl groups, right? So we're going to remove this guy, and we're going to get a double bond between the 2 carbons. Then once we have that double bond between the 2 carbons, we can undergo a hydration reaction, and we can actually create the isocitrate. So it's really just preparing the molecule for the next steps in the reaction. Okay. So the next one, Isocitrate Dehydrogenase. This one is, again, very favorable, very likely irreversible, right? Irreversible. Now, as we say these things, they're irreversible and those kind of things, it's actually really hard to measure delta G values inside of the mitochondria. That's a really big challenge. So we say they are, and we have pretty good estimates of what they are, but it's really hard to get an actual value. So this is another rate limiting step, just like we talked about in glycolysis, right? And so this is highly regulated. This step is a very highly regulated step. And so if we look, we're going to regulate this using ADP. We're going to regulate this using calcium, right? ATP is going to be a regulator. NADPH is going to, not P, NADH is going to be a regulator. So remember, your ATP and your NADH, these are products of these reactions. So these are going to be negative effectors, right? Whereas your ADP and calcium 2+ are going to be positive effectors. So those are negative, ADP, Ca2+ are positive. So if we look, this is an oxidative decarboxylation. So if we look at isocitrate, what's happening in isocitrate is that we are losing 2 hydrogens, right? So we're losing a hydrogen here, we're losing a hydrogen here, and we're going to convert that into transfer those hydrogens to our electron carrying molecules of NAD+. And so when we look at this, we're actually going to make a transient intermediate. So this very first step, the only thing that we are doing here is oxidation. And it's our second step where we go from oxalosuccinate to alpha-ketoglutarate, where we actually have our decarboxylation. So remember in the beginning, I told you it was an oxidative decarboxylation reaction. It's actually a 2-step process, right? And so in our second step, what we're actually doing is we are losing this carbon right here. This is the carbon that we're eventually going to lose, and we're going to lose it as CO2. So that's why we have that step, right? So this is really important because when we make alpha-ketoglutarate, alpha-ketoglutarate is also a key shared metabolite between the citrate cycle and lots of other pathways. And so it's, for example, a substrate and a product in amino acid metabolism, right? In all kinds of different things. So this is a place in the citrate cycle where products can go in and products can come out. And you have to remember that the citrate cycle is a hub, right? And so lots of things can come into the hub. It's not just sugars from glycolysis. So you just have to kind of remember that. All right. So now our second oxidative decarboxylation reaction. So this is oxidative decarboxylation, right? So in this oxidative decarboxylation reaction, we have alpha-ketoglutarate dehydrogenase complex. Now, this is a complex that is really similar to our pyruvate dehydrogenase complex. It's the same catalytic mechanism. So just like in our pyruvate dehydrogenase complex, what are we going to produce? The same things. We're going to produce a CO2 molecule. We're going to use CoA in order to be a carrier for our carbon-containing molecules. So we're going to use up a CoA, and we're going to convert NAD plus to NADH. And we're going to end up with a substrate now, in this case, that has 4 carbons attached to a CoA molecule instead of 2 in the previous. But it's the same thing where this is also right, a very high negative delta G reaction. So this is another irreversible reaction, right? So this one is another good point for regulation. So this one is also regulated by energy charge of the cell. So regulated by energy charge of the cell. And so you already know this mechanism. This is a mechanism that you've already really learned because it all has the same E1, E2, E3 complex. So this one looks complex, but it's actually really easy. So now that we have Succinyl Coa, right, we're going to convert our Succinyl Coa to succinate. So this is done with Succinyl Coa synthase, and it is a substrate level phosphorylation. So remember, in substrate level phosphorylation, you are actually going to create those energy carrying molecules, right? And so very easily, once we make, in this case, GTP, the GTP has a reaction with a DP, right? And we're going to make GDP and ATP, right? And so the delta G value for this is basically 0. So it doesn't require energy to actually go, right? So in order to do this reaction, right, you're going to have several high energy intermediates that are formed to make sure that energy gained by the cleavage of our thioester bond, right, is not lost before being used to generate our GTP. So it's kind of basically like pass the hot potato to move the energy required to make the high energy molecule. So if we're changing all those bonds in those high energy intermediates in this reaction, by the time we're done, we have something that's an energy carrying molecule of GTP. So next we have our Succinate Dehydrogenase, right? So this is reaction 6. This links our citrate cycle to the electron transport chain via a different electron carrier. So this one actually produces FADH2, right? So in this one, we have this FAD that's covalently linked to the enzyme Succinate Dehydrogenase. And so this is a protein, the Succinate Dehydrogenase is actually a protein that is associated with the inner mitochondrial membrane. It's on there. With inner, it's going to be important later, mitochondrial membrane. I'm going to say, remember when, right? So the whole point of our succinate dehydrogenase reaction is to transfer that pair of electrons, right, and 2 protons to FAD. And so later, we're going to pass these 2 electrons from FADH2 here to the electron carrier coenzyme Q that's actually in the electron transport chain. Now, you have to remember that FADH2 and NADH go to different places within the electron transport chain. So you're going to derive different amounts of ATP from FADH2 than from NADH. So we're gonna have to be kind of careful about that. When we get to the end, we're gonna quantify how much ATP we actually use, how much ATP we actually make, excuse me. So now we've got fumarate, right? So once we make fumarate, what we're going to have is a hydration reaction. And in that hydration reaction, we're going to convert fumarate to malate. Now, it's actually a 2-step reaction. And so in this process, right, we're gonna end up with a carbon ion intermediate. And in this reaction, we're actually going to produce the L isomer of malate. Usually we're producing D, but in this case, we're producing L. But it's kind of interesting. Okay, so then we move on and we're going to convert malate to oxaloacetate. So oxaloacetate, remember this is the end of the cycle. Reaction 8, we've made oxaloacetate. And remember in these last couple of reactions, we haven't actually changed the number of carbons. We've kind of rearranged the molecule, but we haven't changed the number of carbons. So this is going to oxidize the hydroxyl group over here. So we're going to lose a hydrogen here. We're going to lose a hydrogen here. We're going to make NADH. So this is another dehydrogenase. That's how you can kind of recognize which reactions these are, right? And so we can see that these reactions where we have oxidative processes occurring, we're making those NADH electron carrying molecules. So then we're going to use our oxaloacetate in the next loop of our cycle, right? Okay, so next section is to talk about regulation. So when you're studying this material, it's kind of important to be able to answer questions about it. And you can use those end of section questions. Those are really good. But also kind of think about these reactions and which reactions are similar to one another, which reactions are doing kind of the important work. So in this case, let's talk about which reactions catalyze oxidative decarboxylation of our pyruvate to acetyl CoA to generate our 3 CO2 and 3 NADHs, right? So if you look at the first one actually doesn't occur in citrate, right? Our first one actually occurs before the citrate cycle. So we're going to go from pyruvate to acetyl CoA, right? So which enzyme does this? We're making CO2, we're making NADH and an H+. So which reaction? This is our pyruvate dehydrogenase. And it's a complex, but it's not just one enzyme. So that's one of them. Now, when we go directly into the citrate cycle, now we have 2 more oxidative decarboxylation reaction. We have when we convert isocitrate to our alpha-ketoglutarate. We're going to make CO2, we're going to make NADH, and we're going to make H+, right? So that's isocitrate. And you think about these names actually make sense. Isocitrate dehydrogenase. And then our last one, we're going from the next step, alpha-ketoglutarate, into making Succinyl Coa. CoA, right? And we're going to make, again, CO2, and we're going to make NADH. Okay, so this one is alpha-ketoglutarate, And remember, it's a dehydrogenase. So all three of the reactions that are occurring where we have oxidative decarboxylation, we have dehydrogenases. So that's really, really important to kind of recognize that we have similar enzymes that are doing similar processes and they're having similar results. So when you're looking at these reactions, if you learn the names, you're actually learning what's actually happening, because you know, if it's a dehydrogenase, what are you doing? Oh, it's an oxidative decarboxylation reaction, right? And these occur very early on. These are where we're pulling off carbons, right? So where did those 3 carbons come from? Those 3 carbons came from glucose that got cut in half, right? So we had a 6 carbon molecule, we cut it in half, we have 2 3 carbon molecules. Now those 3 carbon molecules, when they go through the citrate cycle, it takes 2 cycles to get rid of all of them. But eventually each of the 3 is going to be released as carbon dioxide. And so you end up using up the carbon containing molecules and making simply electron carrying molecules. So that's kind of the basics of the citrate cycle. So next time we'll talk about regulation.